Here are the key players of DNA replication. The double-stranded DNA is split into two strands using the helicase enzyme. This creates a replication fork. The primase enzyme synthesizes RNA primers to start the replication process. The DNA polymerase III synthesizes DNA from the RNA primer start. The enzyme topoisomerase then relieves the strain on the twisted DNA to ensure that the bonds don't break. This ends the DNA replication in the leading strand. Now, in the lagging strand, the enzyme primase synthesizes the RNA strand and begins the DNA replication process. The DNA polymerase III now replicates the DNA. Since this happens in the opposite direction, more double-bonded DNA are being broken by helicase to single DNA strands. The enzyme primase goes back to these open templates and begins the RNA synthesis. In the same way, the enzyme DNA polymerase III comes and replicates the DNA here too. Though inaccurately represented here, there is usually a break between the RNA formed and the DNA synthesized. This break is joined together by DNA ligase. These separate sections of RNA and DNA are called the Okazaki fragments. The RNA primer is now removed by the DNA polymerase I. This replication process happens in the replication bubble and is repeated until the DNA strand is fully replicated.